Hello, everyone. Thank you for joining us. We will be discussing our presentation on toxic cyanobacteria identification. Our group members include Natalie Kirkbride, Emily Jones, Vicente Sanchez, and Lisa Zavala. We are under the guidance of Research Advisor Dr. Lucia Vasquez under the Department of Biology.
Hello, my name is Emily Jones, and I'm going to get us started here with the background information regarding cyanobacteria and algal blooms and their effects on water quality, as well as current screening practices. So the focus of our research is cyanobacteria and the toxic algal blooms that cyanobacteria produces in freshwater systems. These toxic algal blooms can result in illness and mortality in humans, wildlife, pets, and livestock. In recent times, actually, the Environmental Protection Agency has issued a health advisory as a result of these toxic algal blooms, which has also resulted in the World Health Organization requesting species identification of these cyanobacteria genera in order to further identify them and be able to clean out our freshwater sources. So the recent and current practices for cyanobacteria identification is actually the method of morphological identification, which has shown to be inadequate for cyanobacteria. Not only is morphological identification expensive and time consuming, but it's also inaccurate at the species level. Morphological identification depends on looking at aspects such as cell type, type of fission, shape, and size of the cell, which can be pretty tricky with cyanobacteria considering a lot of species look very similar. So a good way of deciphering these similar species is DNA barcoding, since each of these species will have their own unique DNA barcodes. The particular species we'll be looking at are species that have high levels of toxic algal blooms in freshwater. These species include Microcystis, Cylindrospermum, Dockilospermum, and Planktothrix. And we'll be looking at these for our research. So the objectives of our research is to determine a suitable DNA barcode using the ITS region for species within the cyanobacteria genera of, again, Planktothrix, Microcystis, Dockilospermum, and Cylindrospermum. And we're going to do this by testing the utility of the ITS by examining sequence variation. These genera are some of the most problematic cyanobacteria, again, due to their toxic algal blooms. So identifying these and creating these barcodes would actually allow us to identify them in freshwater sources and aid in the implementation of management practices.
Hi, my name is Vinny Sanchez, and I'll be discussing our research plan and what we've accomplished so far. So as you can see, here's our research plan as a flowchart. In its most simplest forms, our research involves 9 steps. Currently, we are in preparation for sequencing, DNA sequencing using Sanger sequencing. Our samples were obtained from Carolina Biological Supply Company. One thing I would note is that here, Oscillatoria is the new name for the Planktothrix genera, and Anabena is the new name for the Dockilospermum genera. And as I had mentioned previously, we are in part of our research process includes DNA isolation. We actually use the Saba et al. Methodology. And this is because it actually had an easier lysis approach. It works on filamentous bacteria, and its protocol actually specifically mentioned the ITS gene, one of our target sequences. Now onto our DNA isolation results. Here is our gel, and here are our results in a 1% gel. And as you can see, there was high quality DNA extracted from the genera Cylindrospermum, Microcystis, and Oscillatoria. However, preliminary isolation resulted in insufficient or low quality and DNA from Anabena.
So the primers were designed by downloading the internal transcribed sequence, spacer sequence from GenBank. The DNA sequence alignment was done in the program Muscle to find any variation among the species. If they were present, the primer was then designed using PrimerBlast on NCBI. As you can see, this is one of the primer sequence examples that was used for microcystis. So the ITS region amplification was done for these cyanobacteria through temperature gradient PCR. The ones that didn't show as much data, we had to redo with PCR product reamplification. And then here are the temperature gradient PCR results. As you can see in figure 7, the top row contains cylindrospermum samples, while the bottom row contains Anabaena samples. While figure 8 has the microcystis samples, while the bottom row contains Acceleratoria samples. What we determined from the temperature gradient PCR results was amplification was found for cylindrospermum and microcystis. Anabaena and Acceleratoria did amplify, but had low amounts of product. And these are the ideal annealing temperatures for cyanobacteria. So cylindrospermum had 53, Anabaena 47, microcystis 53, and Acceleratoria 48. PCR product reamplification, Anabaena did not apply, but had another target sequence amplified. Acceleratoria did not amplify. So here we have figure 9, the agarose gel for PCR product. Figure 9, lane 1 has the DNA ladder, while lane 2 had Anabaena, and lane 3 had Acceleratoria.
So after obtaining the ample PCR products, our next move was then to clean the samples. And because only Oscillatoria and Anabaena didn't produce enough PCR product to sequence and therefore clean, our next move with those is to run temperature gradient PCR again. After we obtain ample PCR product, we're then going to exosap it, which will remove any remaining nucleotides and primers. And then we will prepare sequencing reactions for both these samples using Sanger sequencing methodology. Because cylindrosperm and microcystis have been cleaned and did show ample product, they have been cleaned and will undergo sequencing reactions. And therefore, once we get those back, they will then be analyzed for interspecific variation. If the sequenced ITS regions show higher interspecific than intraspecific variation and are distinctive for each of the species, then the barcode development would be concluded as successful. Then once it's concluded as successful, we will be able to test this on the EPA contaminated sample and determine if the sample has any of the species that we are looking at and have created DNA barcodes for. Thank you for watching our preview, and we look forward to answering any questions during our designated time slot on Friday. Additionally, here is the literature cited throughout the presentation.